thank you for attending our Grand Rounds lecture. I'm pleased to introduce to you our speaker, Dr. Saul Karpen. Dr. Karpen is the Chief Scientific Officer for the Stravitz-Sanyal Institute for Liver Disease and Metabolic Health at VCU. He's a professor of internal medicine and professor in pediatric medicine. He holds the Philip Heilman Endowed Chair of Medicine and Microbiology and is a pediatric hepatologist. The aim of his research is to understand the root causes and adaptive mechanisms for cholestasis, including recent work focused on discovering novel genetic causes of biliary atresia. His overall goals are to develop integrated programs to improve clinical care, enhance basic and applied clinical research, including the discovery of new therapeutic agents focused upon the unique and pressing needs for infants and children with liver disease. On a national and international basis, he's an active participant in 2 National Institute for Diabetes, Digestive, and Kidney Disease supported consortia for liver disease. Dr. Karpen is currently the Senior Counselor of the American Association for the Study of Liver Diseases and is the upcoming President of the Society in 2026. We're grateful for Dr. Karpen's expertise on this topic and look forward to learning from him. At the end of the lecture, we'll leave time for questions. Please submit questions via the chat function on Zoom as they arise during the talk. To claim CME credit for today's session, please only text the code shown at the end of the presentation to 804-625-4041. I'll now turn it over to Dr. Karpen.
Wonderful. Thank you so much, Dr. Shaik, and thank you, Greg, for letting me talk. This is new to me. Part of this is kind of a group get to know everybody tour. Since I've only been here since March, I can give you kind of a very brief addition to what you just heard. I am a pediatric hepatologist, but I really like adult medicine. I'm learning more each and every day. I like collaborating. My role here, and I'll go to the next slide, is a new role as Chief Scientific Officer. And I give strong credit to Dr. Stravitz-Sanyal for creating a liver institute that in our world, which is, unlike oncology, neurology, and cardiology, there are really no large privately funded institutes in the world that are going to study liver disease. This is actually the biggest. So this gift of over $100 million really is changing the face of how we approach liver disease, both locally and globally. So things are new. If I gave this talk in a year, you can start to see what cores we have developed, but I'd love to meet with people. We are developing a number of different cores that are going to be both metabolic in nature, but also multi-organ in nature. Obviously, we're going to be talking about fatty liver issues, but I'm going to talk today about really what my personal passion is, which is really figuring out what is the #1 issue in pediatric liver disease, and that is the causes and the treatments and the interventions for biliary atresia. And I thought that when I was going to talk about it, I'd really give a framework that may or may not resonate with everybody in cardiology, but for those of you who have to take your boards, you're going to have to know about heterotaxic liver disease and cardiac disease, and I'm sure you run across it. But biliary atresia is what I would consider to be a heterotaxic condition, and we're using that to make our discoveries to figure out what causes the disease. So I have a couple topics today. I will do my best to give about 15 minutes at the end. I'm going to talk about BA, talk about what we don't know. I'm not going to give you the whole lecture about the fact that we are still, for our -- disease, not knowing what are the causes, and I'm really chipping away at genetic predispositions. We have discovered a gene and have created a mouse where it's loss of function of one gene, this PKD1L1, which was actually first discovered as a cause of heterotaxic cardiac disease, is a cause, and the first genetic cause found for biliary atresia. Through a national consortium, NIH funded, we are discovering new genes, and I run that. But I also wanted to switch gears a bit, because as a clinician, and I worked in a number of different places, some that were in the pediatric world, quite large, where we saw a lot of liver disease, but also the ramifications in other organs. One of the things we found was that there really was a cardiac phenotype to cholestatic liver disease. And so that is our condition in pediatrics that we're calling cirrhotic cardiomyopathy, slightly different, but also similar in concept as far as I understand it, to what you would see. But we have found that there's a role for circulating bile acids affecting the heart and the metabolism of the heart. I'm going to touch briefly on that and really highlight my colleague's work, who was my mentee when I was in Baylor, who has continued this work. All right, let me go quickly about biliary atresia, and please feel free to interrupt. I don't know if I can see chats, but let me know if you want to know more. I'm going to go very brief. Biliary atresia, and what I'm showing here with the graphics is what most people understand is the obstruction and destruction of the extrahepatic biliary tree. And on the right-hand side is a histologic slice of the extrahepatic biliary tree, where you see denuding of the endothelium, the lining, the cholangiocytes, and this incredibly rich fibro-inflammatory infiltrate. This is the histology at 3 months of age. I mean, it is this unbelievably fibrotic disease. It is the most fibrotic disease in liver, full stop, where you can have end-stage liver disease by 3 to 6 months of age, all with this exuberant fibroinflammatory infiltrate that's centered around the bile ducts. But believe it or not, even though it's the #1 etiology for serious liver disease in infancy, it's the #1 reason to transplant a child. It's also the #1 reason to do any solid organ transplant in a child. In other words, if you're less than 1 year of age and you need a solid organ transplant, it's for this disease. And yet we know very little about it. So that just irks the heck out of me. And that's why I am working on, and I believe we're on the discovery end of figuring out what some of the causes are. Okay. But one of the things that gave us a hint as to perhaps there's some group of genes or group of, I would say, features of development that go awry where the bile ducts don't necessarily develop, and this is pan-biliary, is that about 10% of these kids have heterotaxic conditions, and it's considered biliary atresia with splenic malformation. Most of the time it's polysplenia. A number of these kids have left atrial isomerism defects, and a number of them have situs defects. So when you start putting this together, it's the first thing that comes to everybody's word association brain is ciliopathy. And so us too, but the group of conditions where ciliopathies have really been well explored, especially the complexities of it, is in congenital heart disease. And I actually use the genetics of congenital heart disease as my lodestar, frankly, to figure out what's going on in the complexities during development of biliary atresia. So we use this group. Now, when you have a rare disease, each institution doesn't accrue enough patients. So this has to be the purvey of a multi-center consortium. And luckily, a bit over 20 years ago, the NIDDK supported the development of a consortium to study pediatric cholestatic liver disease, of which biliary atresia is the centerpiece of it. And I'll talk about that. And this is the way in the modern era now with gene sequencing and gene sequence analyses, we can start to tease apart some of these complexities. Okay, so what do we know about BASM, or biliary atresia splenic malformation? As I mentioned, pretty much everybody has a splenic malformation, but also a bit over a third have situs inversus and other vascular abnormalities. About half or so have intestinal malrotation, absent IVCs. Almost half have some degree of a congenital cardiac anomaly. Not all of these are surgical, as I'm showing you on the right-hand side. This X-ray is what we can do in pediatrics. You know, take a picture of the whole chest and abdomen in one go. This is post-surgical. You see this is someone who has a heterotaxic heart condition, midline liver, malrotation, and here you can tell from the NG tube that the stomach is over here. And so this is a group of patients that these are the ones that we'd want to say, if there is a genetic drive to biliary atresia, it would be in this group of patients most obviously, especially if it's more monogenic in nature. And that's really where our brains are when we start thinking about genes associated with disease, is to try to find those single gene defects. So working on this premise, we started to identify, well, how much of this, what are the clinical manifestations? The NIDDK consortium acronym is CHILDREN, so it's the Childhood Liver Disease Research Network. And as I mentioned, it's been going on for 20 years. I transferred this from where I was recently, which is Emory, and then now it's firmly here at VCU. So I'm one of the PIs. And about almost 10 years ago, we collected a group of patients that had these laterality defects and tried to figure out how much and what percent of those with laterality defects also had cardiovascular, pulmonary, GI, et cetera. And as you can see, cardiovascular defects are quite prominent in those where from a liver brain, we're also seeing that there are cardiac heterotaxy events, okay? Well, what about the other way around? What about from your world, okay? From the cardiac world, how many of your patients who have left LAI will also have biliary atresia, and how important is this? And it ends up being that it's about 10% or more, sorry. It's about 10% or more that have biliary atresia. And it ends up being that outside of poor outcomes due to the cardiac disease, this is the second most common reason for these kids to die or have a serious adverse clinical outcome. So these are somewhat together. And again, from a gene discovery standpoint, there has to be some overlap, okay? This can be determined prenatally. So obviously with the prenatal echoes that are done to determine congenital heart disease, one can also look at the gallbladder on these kids. And it ends up being that roughly same idea, 7 out of about 105 of these live-born kids with heterotaxy actually had defined biliary atresia. So it is something that does go together, but certainly not everybody, but one that one has to look for. This is not something you in the adult world ever have to worry about if it wasn't discovered early in life, because this is a developmental defect. Okay. Very recently, if you are interested in the genetics of congenital heart disease, this is the most recent review from who I would consider to be one of the great leaders in the field, Dr. Cecilia Lowe from Pittsburgh. And this is an annual reviews paper just out in May. Where roughly speaking, just so you know, congenital heart disease is about 1% of live births, and about 10% of the patients with congenital heart disease seems to have, well, if you look for it, a genetic cause. And so it lumps into a few categories, either those that are involved with the formation or function of cilia, or some transcriptional regulations or cell signaling. So there's a large number of gene defects that may lead to the abnormal looping that goes on with the development of the heart, and a similar idea with the tube system in the liver. Okay. I'm going to highlight this gene, as I already briefly mentioned, called PKD101, which is on the cilia, and works in concert with PKD2. All right. So the way I look at BA with heterotaxy is that really this is all part of a spectrum of development that I find quite exciting. I think it's a bit frustrating when we are all trained to think about monogenic causes or some simple polygenic causes. There's something different that often can go on during development, where on certain days of development, when the heart is developing, if the environment is one with too much glucose or some certain extra toxin or something that's supposed to be responsive to a signal doesn't get that signal, then you have some downstream consequences that may be complex to find. But there has to be some genetic contribution, especially when you have both BA and heterotaxy together. So I like to think of BA as a cholangio, meaning bile duct ciliopathy. And that makes sense when you start thinking about looping and tube systems, okay? So why is that? Well, cholangiocytes have a single long primary cilium that floats in the bile. So it's a sensor mentality. So anything that can affect cilia may affect bile duct development. And what I'm showing here on the left is just a graphic representation that these cilia are just floating in as sensors and purveyors of signaling related to bile. So there is, I think, on the hand wave side, some type of degree that perhaps there is a ciliopathy component to this. And again, I think the heterotaxic conditions make people a little bit more comfortable with this approach. Okay. So what did we do? And this is a published work that I'll go quickly with. We said, look, we just need enough patients. Exome sequencing is coming in. And by the time we were able to do this and get enough money, we did some straightforward things, whole exome sequencing on what at that time was just 67 kids. And we focused, because this is the first time anyone's ever done this, on a little over 2,000 ciliary and biliary atresia candidate genes. And for me, I just had a pretty easy way to think about this. They need to be rare. It's a rare disease, about 1 in 10,000 births. It has to be both alleles. The genetic causes have to be damaging with our variant analyses, and it just has to make sense biologically. And so the functions of these genes that seem to make more sense, those that are involved with left-right determinations, ciliary function, and as well as has to be expressed in cholangiocytes. All right. And this came out with, when we looked at these 67, 5 of these kids had quite strong biallelic damaging variants in PKD 101. And this was published in 2019. So what is this gene? Well, it's in the PKD gene family. On the left-hand side is PKD1, also known as polycystin 1. It's a gigantic gene, about 5,000 amino acids. This is only 2,800 amino acids, okay? Still another gigantic gene. And it works in concert with polycystin 2 or PKD2 to elicit some type of sensing and calcium signaling at varying times of development. And in fact, PKD 101, PKD2 interactions are the first interactions that determine sidedness in the developing node that early in development. Later on, it's involved in cardiac development and, as I'm showing you, bile duct development. So these are signaling molecules. Okay. So here's a paper that I'm sharing for you that came out in 2016, where the first group, this came from Baylor, found that with patients with congenital heart disease, they did whole exome sequencing. This is one of the exome sequencing centers, that these 2 different patients had chain terminating variants in a gene that was not on the radar screen for congenital heart disease, PKD 101. So this is definitely a gene that is not superfluous. One needs this gene fully functioning to develop heart. These 2 patients had quite serious congenital heart disease. You can see just from the x-rays, etc. Okay. So what do we know about it before we started to do some work? Well, if you make a mouse that is fully deficient in PKD 101, it's mostly embryonic lethal. Those that can make it to birth, about 50% of them have heterotaxy. So there is almost a randomness of, when you don't have PKD 101, which side your stomach's going to be on and whether or not you're going to have serious congenital heart disease. So since we can't use the full knockout mouse, we just made a tissue-specific mouse where we knocked out PKD 101 in developing hepatoblast. And what happens in developing liver is that the primordial liver cells become either hepatocytes or, in 5% of the time, cholangiocytes. But they both come from the same cell primordium. So we were developing a model where we can knock out PKD 101 only in these developing hepatoblasts. And the way you do that is quite straightforward. You knock it out with a gene called alpha-fetoprotein, which is expressed before albumin, around about day 11 or so of development. And so we created a floxed version. And what I'm showing you on the lower right-hand corner is this fabulous grad student I had. I will never have a grad student, I think, who is this talented and productive, Dominic Callan, who's now just started his postdoc in a, I would say, organoid lab at MIT. And so made a floxed version of it. It creates a chain terminating section only in AFP expressing cells, so only in hepatoblasts. And what happens? Well, what happens is that we end up with a mouse that has more fibrosis around the bile ducts. This is a serious red stain around the bile ducts. Here's a portal vein system. More cholangiocytes, more inflammatory cells. In addition, about 50% of the cells should have cilia. This is a scanning electron micrograph of the flocks, of the wild type, and this is the knockout version. About 50% don't have cilia. So I'm showing you the most dramatic SEM pictures, but just to share with you really that there is a need for cilia. That was a mild phenotype. So we asked the question because alpha-fetoprotein is not expressed in the extrahepatic bile ducts. The way that we can create obstruction is with bile duct ligation. So we just asked the question, is the knockout mouse worse off than the wild type mouse in the setting of obstruction? And the answer is an exuberant yes. Wild type on the left, knockout on the right, and you can easily see, and we quantitated this in whole slide scanning, a much more exuberant fibro-inflammatory infiltrate in the knockout compared to the wild type. And this is just showing the quantitations of fibrosis, number of cells, etc. In addition, we saw that these cholangiocytes, the bile duct cells, were much more reactive. So remember, PKD101 is missing in these cells here and eliciting this response. So why? And that's the fun part, right? That's why we do science. Why is this really happening? And so why are they intrinsically more dysfunctional and more reactive? And that's what we're working on now. And I'm showing you a postdoc, is a German gastroenterologist who was in the lab, who was able to make organoids. And so the very straightforward question here is, if these cholangiocytes that lack PKD101 are more reactive, we should be able to model this in vivo and in vitro. So isolating the cholangiocytes from wild type on the top and knockout on the bottom, they were definitely different organoids. We couldn't make these nice round organoids as you see here when PKD101 was knocked out, okay? What else happens? They're just totally disordered, right? Knockouts don't look good. They make more chemokines and cytokines, and I'm just showing a couple of these just to lay the flavor of where we're going with our work here. So if those of you are looking for any sort of fibrosis in heart muscle, we would really love to collaborate because we're developing a fibrotic, basically a fibrotic core, both in live animal imaging and also in humans. So we'd really love to talk about this. And that's part of my role as the CSO for the Liver Institute. So this is our mouse model, and we're going to start to do towards the end of this year, beginning of next year, working on these live animal imagings. Okay. So what about the fact, are there any other BA genes? And we can't do that with 67 patients, and we can barely do it with what we've collected. But after 20 years or so, we collected a number of BA patients. We now have over 2,000 in the consortium, and we got enough money. I was able to convince NIH to pay for this, to do exome sequencing on them. So make a long story short, we have exome sequencing that we're still, this has not yet been published, and we're finishing this up, and hopefully we'll publish this by the end of this year by working here at VCU, of 1,700 DNA samples. This is now getting to the range where we can start to do perhaps some polygenic approaches, or at least find some that we can then screen just like we screened for PKD101. And about 10% of these are BASMs. But as for those of you who've ever done gene hunting, you get a ton of variants, right? And so do similar sequencing filters where high CAD scores, rare, low mean allele frequency, keep if we have two, et cetera, et cetera, et cetera. We get to a list of genes, and now we're going through the processes, not just being focused on, let's say, ciliopathy genes, but being a bit more open and unbiased to look for other genes. And we're doing this from an autosomal recessive and an autosomal dominant approach. This takes time. Well, we can't take a gene, make a mouse, wait a year, decide whether or not it's going to work, okay? So what do? Well, something pretty simple. The hypothesis here is one where we believe that it's loss of function that leads to a change in function of the cholangiocytes. So a very simple approach. One can take mouse cholangiocytes, and we will do this with human cholangiocytes in the next number of months. Culture them, make organoids, either from the extrahepatic tree or the intrahepatic tree, and just take the candidate genes of interest, knock them down with a lentivirus, and see what happens. And if we have an altered cholangiocyte function, then we know we have a gene that, if improperly functioning during development, can lead to maldevelopment of the bile ducts and what we see as this phenotype of biliary atresia. And this is what Dominic was doing, and we're going to do it in the lab here, just starting that up this week, actually, and giving you some hints that some of the genes that have more variants than they should, we started to knock them down. And what I'm showing you here are a couple of positive and negative controls. Positive controls, if you knock down PKD2, you get cystic biliary disease, just like you get cystic renal disease, where we get cystic ducts cells in vivo. And if we knock down some of these ciliopathy genes that are more highly represented with variants in our data set than there should be, we actually get abnormally developing and functioning cholangiocytes. So this we can do in 2 weeks. And so we can plow through a whole bunch of candidate genes. So if there's anyone who's interested in ciliopathies, anyone interested in reactive cholangiocytes or reactive cells that are in any tissue, we'd love to collaborate and discuss our plans, okay? So biliary atresia was this disease, and I believe we're really changing the discussions on BA from thinking it's just some random, perhaps a virus, perhaps a toxin. It's really just maldevelopment. And so we're finding genes that are leading to that, again, using congenital heart disease as our guide. And what I'm showing here is that normally you have a nice tight junctions in the bile ducts, but in the setting of biliary atresia phenotype, these are abnormal. The cells are abnormal. They secrete all these chemokines and cytokines and bring harm to themselves. And so we're working on this, okay? And in the interest of time, I want to get to the next step, and I'm going to skip this. And really want to talk about something cardiac that I started when I was at Baylor, and have really worked nicely with this really fantastic critical care physician at Texas Children's Hospital. I don't know if any of you have ever been to the Houston Medical Center, but it's a square mile of medical buildings. The Texas Children's Hospital is, I think, 22 stories in about 4 buildings, and 4 levels of that hospital is ICUs. And one corner of that is a liver ICU that Dr. Desai, Morishwar Desai, shown here, is running. And so he worked in my lab when I was at Baylor, and we really worked together because we saw a patient, and we saw this patient. 9-month-old, BA, end-stage liver disease, on the liver transplant list, came to the ICU and profound with fever and hypotension, despite all the inotropic supports, really poor cardiac output. These are always the danger signs, as you well know. You see someone who should be responding with tachycardia, and they're not. And you know, in kids, you respond with increasing your heart rate primarily. Nothing happened, and the patient died. And so this was not part of our lexicon to think, why did these patients die like this? And why was there an inappropriate cardiovascular response to sepsis? And why was there really an inadequate response to pressors? And I'm going to be very brief in the interest of time to just give you a few snippets of where we think that there's a response of the heart to prolonged cholestasis that makes the hearts of these kids inadequate responders to these stressors. And that's where we have this concept of cirrhotic cardiomyopathy. And the way that we looked at this, and this is a paper from over 10 years ago, where we really just looked at the structural and functional alterations in kids in exactly this setup, not this profound where they come into the ICU in an awful state, but waiting for a liver. Are their hearts any different? And very simple approach, just doing 2-dimensional cardiac echoes. often we do these echoes to look for the consequences of perhaps heterotaxy before they get to the OR for their liver transplant. But we're also looking at now the cardiac muscle and the cardiac function. And so they just really had 2D echoes. And the bottom line is there are differences. The hearts are bigger, they're less, their relaxation in diastole is different from other kids. And so this is where we get to this definition of cirrhotic cardiomyopathy. And these are slides from Morishwar. And I just would love, encourage you if there are some interest here with cirrhotic cardiomyopathy that we can reach out to him. It's really a blunted contractile response to stress, altered diastolic relaxation, and there really are some EP abnormalities without any known cardiac disease. But why is this, right? It's not a gene issue. It's a heart responding to the milieu of a cholestatic patient. And so there are probably mediators that just trickle out of this damaged liver, where the first stop is obviously going to be the heart, as well as there being a number of circulating molecules that the heart sees because the liver is not able to perform all its functions. Okay? And what does one see? One sees a number of different things. The number one thing that at least I can pay attention to, and I would love if there's somebody here who is interested in this biology and physiology, that the LVMIs are higher and that the hearts really do get big. We've been able to model this in mice. If we make a cholestatic model in a mouse where we don't let bile flow, the hearts of these mice get bigger and don't function as well. So there is an actual model that if one looks above the diaphragm as a liver doc, you actually see some quite interesting and important physiology, okay? And so what happens to these kids? And this is a paper that was published a couple of years ago from the group at Baylor and Texas Children's to look at what happens. And does the cardiomyopathy resolve after liver transplant? It seems to. As many things in medicine, things get better in pediatrics before they get better in adults, okay? And that, is this LVMI index associated with consequences? And the answer is what we think is yes. And this, if one looks at multi-organ dysfunctions before and post-transplant and death, anytime you have a high LVMI, you have worse peri-transplant outcomes, worse pre-transplant outcomes, worse post-transplant outcomes. So the patient is worse off when there are features of cirrhotic cardiomyopathy than not. Okay. I mentioned modeling this in mice, and these are papers that have been published, but I want to show you some cool things. If you do MRs on these patients, DDC is this cholestatic molecule that we put in the feed that blocks bile ducts, that the hearts get thicker in these mice within about 2 to 4 weeks. And as I mentioned, they are much more dysfunctional. And we also have a story that if we isolate neonatal cardiomyocytes and expose them to higher and higher concentrations of circulating bile acids, that they engage in a hypertrophic pathway where myosin gene expression is increased. So there is a real cardiac feature here that is just waiting to be explored. Morishwar has unfortunately too much clinical work to do this lab work anymore, but I'm really happy because we're going to be making cholestatic mice with their different biliary atresia gene mutations. I would love to have somebody who wants to work on the hearts. Okay. Lastly, from a clinical standpoint, I had a pediatric cardiac fellow saying, well, look, we know that when the LVMIs get high, that there are some adverse consequences. When does the LVMI increase? And if the LVMI is high earlier in life, does this portend a worse outcome? And it ends up being that even at the time of Kasai operation, which is roughly speaking about 2 months of age, we were doing cardiac echoes at that time. If at that early age, not knowing what their future is going to be like, the operation is one where you have a draining procedure from the liver, it works about 50% of the time. Can you predict outcomes based on LVMI? It ends up being you can. This is a small single center study. This was done at CHOA at Emory, that the LVMI at the time of Kasai, meaning 2 months of age, if it's high, predicts a poorer outcome. Okay. So Morris Rohr has really been a lead on this, that this concept of cholecardia, meaning that the heart responding to circulating bile acids. I would love to talk to my colleagues here, and I'm going to start these conversations with my adult hepatologist. There's 200 liver transplants done here a year. There's a lot of end-stage liver disease that is primarily from FAASLD as well as from alcohol, but there's definitely changes in heart structure. And this would be something really important, I think, that there could be great collaborations between cardiology and hepatology to study this concept. So the heart is a target. It really exists in infants. It likely contributes to pre-transplant mortality and perioperative dysfunction. That there are some really interesting, I think, features of cardiac muscle and sensitivities that we can explore in a variety of in vivo models that I think would really, improve the health of these patients. Remember that these patients with end-stage liver disease, they don't just have liver disease and consequences. They also have kidney disease, and they have altered cardiac function. So there's a number of axes that are disrupted. And I think anything that we could do to help these patients will definitely help them, especially those that are on the docket to get transplanted. We would certainly want their hearts to be in better working order. That the DDC feeding of mice, it's a nice model for biliary fibrosis, and that we have definable cardiac outcomes. And it's actually reversible, that if you stop the feeding and you allow bile to flow and the liver to become less fibrotic, the heart responds appropriately. So there is a reversibility even in the mouse model. Okay. And as I briefly mentioned, there is bile acid signaling in heart, and it goes through this G protein coupled receptor called TGR5, which is a receptor for bile acids. This is actually the receptor that in the gut makes GLP-1. So there is a known signaling cascades that go on. In the heart, there's activation of various signaling that increases cardiac hypertrophy. And what we haven't really explored well at all is the changes in cardiac cellular metabolism. Okay. I want to thank a few people, and I really am a bit earlier, which is good because there's certainly time for us to talk about the Liver Institute and potential collaborations, but really thank people in my group who are really strong contributors. I want to highlight this person here, Paul Dawson. Paul and I ran a joint lab at Emory. He and I really believe in this concept. He's the person who's cloned a number of the bile acid transporters. And so Paul is still a collaborator. He's still working at Emory. But this concept of being a joint lab is something that's embedded in my heart and brain. And I really like this concept of collaborating and working together. It really is the only way we make discoveries and improve patients' lives. And I'm going to end there, team, and I'm really open to questions and can really talk about what we think we're going to be developing in the Liver Institute that would be of benefit, and hopefully for new collaborations. And Fadi and I have talked a little bit about it, and I think, as I mentioned, in about 6 months in a year, you can start to see some things where there are palpable means to collaborate, both from a core standpoint and also from some animal models. So thank you.
Thank you so much, Dr. Karpen, for joining us this morning and the wonderful talk. Just for everyone, we don't have CME credit approval just yet, so don't send in the text to that number for now. Shannon will send out an email to update you. So Karpen, I suppose one of the questions I had was about the cardiomyopathy in patients with biliary atresia. How does it impact the timing of liver transplant consideration?
Not yet. And we need to do this type of a study. In the consortium that I mentioned, we now follow about 2,000 biliary atresia patients. By the time of a year of age, about half of these will have had a liver transplant. So there's still a number of patients that live with their native liver, and they are 75% of them are cirrhotic, okay? So what is the time that one thinks about either maintaining health, because we are now, we don't have any approved medications that can change the course of what happens next. So it's really being followed, unfortunately, in the worst possible way, which means episodically, and not as a proscripted mention. Those things I mentioned, you know, we're the first people to have discovered this concept, which means that it's just going to take time to cascade this around institutions. And so there are a number of institutions that are incorporating echoes. I think that we should be tracking this because it is a real predictor of poor outcomes, but it's also probably an ancillary predictor of, you know, poor outcomes with other things that are going on, you know, poor growth, ascites, development of GI bleeding. These are things that do happen in infants, but it has not yet. And that is a study that I want to do now that the grant has been refunded for another 5 years, that perhaps doing serial 2D echoes and capturing the information, that we can then use this as for guidelines and guidance. You know, the hard part with pediatric transplant is everybody thinks they're an expert on transplanting, you know, 6 kids a year. You're not, right? And we're not even experts if we are, quote, busy, which is, you know, 2, 3 dozen a year. So putting this all together, we can then really provide some guidance.
Thank you, Dr. Karpen. We'll open the floor to any questions.
Let me see, there's some chat. Okay, great.
I don't see any further questions of Karpen, but we have a couple of comments in the chat box, just thanking you for the great talk and looking forward to any collaboration.
Well, I want to thank you for your time. I know I went quick. I also going to wait to the end to tell about the MOC question, because you may have noticed if you look at the MOC question, it was not something that I initially spoke about. And I took out this slide just in the interest of time, not knowing whether or not there would be a need for it. We have figured out, and what's now standard of care in pediatrics, is that if you have a high direct bilirubin, you need a hepatologist. And I would just encourage you, when you have patients who have high total bilirubins, to fractionate it. And so anybody who's got a high direct bilirubin is somebody who probably needs to see and is followed by a hepatologist. And so that is really the straightforward clinical point I'd like to bring up at the end.
Thank you so much, Dr. Karpen, for your time this morning. We really do appreciate it. There's actually just a question just come in by Dr. McKeer. He's asking if there's any hypertrophy phenotype which stand out of the mice cases you studied.
Yeah, I think it's a great question. From an imaging and from a histologic standpoint, it really is just left ventricular hypertrophy that we found out. But there are also some more subtle, and Marshwar can obviously talk about this better than I can, as far as some of the echocardiographic features that are slightly different. Like I said, there's impaired diastolic relaxation that goes on, among other things. As far as the signaling, a few bits of signaling that have been explored where GSK3 beta has increased, and as I mentioned, myosin heavy chain goes up, expression goes up way high. You know, it's probably 7-fold activated when these cells are exposed to circulating bile acids. So the point being is that when you talk about a phenotype, I think that it's really a response to cholestasis. You know, in a cholestatic liver, and you've seen these patients, right? They have multi-organ issues, but it's not just that bile acids are circulating in the blood. You know, the liver is not doing its filtering and metabolic functioning as well as it should, and these are malnourished patients as well. And the heart responds to this inflammation. It responds to this malnutrition phenotype. It responds to all of these circulating mediators, which many of them are fibroinflammatory. There is more fibrosis in the liver, as Marshwar also has, believe it or not, an autopsy series on patients, because Texas Children's is one of the largest liver transplant programs in pediatrics. And he also sees a fibrotic phenotype in the hearts of those who unfortunately died before they were transplanted. So I don't know if that answers your question, Dr. Makia, but that's the best I can do at the moment. That's why we need help.
Thank you so much, Dr. Karpen. Thanks for joining us this morning.
Thanks, everybody. Look
Thank
forward
you.
to meeting you.